Perfect. Okay, good morning, everybody. How's everyone doing today? All right, we're going to start with David this morning, and he's going to go over some of the background of this. There'll be a number of people on this meeting this morning who didn't join in last time, so we'll go over just basically some of the ground rules that we're going to set, and as well as some of the concept and theory behind why we started this up, and then we'll get to it. So go for it, David.
Yes, hello, good morning, everyone, and thanks so much for joining, especially Europeans who had to get up this morning to join the session. The idea is to have this informal chat about recent Seabird papers, and we felt that this was a good time because probably lots of us have to stay home. It's the fourth week of self-isolation in France, and it sometimes feels like field work on a remote island, except this time the island is our homes. And the question is, hey, where are all the seabirds? That's far less fun than doing field work. But having said that, I think as seabirds we have a long training of getting along with limited resources, limited food, limited water, limited power. So maybe because of this lifelong training, we're all set up for degrowth. So having said that, I will hand over shortly to Grant for some information on how we will run the session.
Thanks, David. Yes, so the plan this morning is basically going to be a, you can sort of consider it like a radio talk show, I guess. So David and I will talk a little bit about the papers, ask some questions to the authors, and then open up the floor to questions. So for those of you who have not used Zoom before, if you open up your chat screen on the right hand side by simply putting your cursor over David or my face, you'll see down at the bottom there'll be a chat and a participants list. If you open the participants list, down at the very bottom you should see a raise your hand option. Now it might be different if you've joined from a mobile device. A mobile device you have to look for it in a different place. When you use the raise your hand option, it'll allow you, I'll be able to see it on my screen, and then I can unmute you. Right now you don't have the ability to unmute yourself. So when I see a hand raised during the question period, then I'll acknowledge you and then bring you on board. The idea here is we want to keep this as light as possible. So if possible, please, if you could limit your questions to simply just one, just because we might have a lot of people and we don't want to keep all of you folks either awake down on the other side of the world or from getting important work done on this side of the world. Yeah, and I think from there we'll start off. David is going to start off introducing the first paper by our Japanese colleagues, and then from there I'll go into the other paper, and then we'll open up to questions as we go. If you do have any issues with Zoom, please message in the group chat. You can message me privately. I'll try to sort you out. As well, feel free to talk amongst yourself on the chat app on the right hand side. That's what it's there for. Be sociable and ask questions to each other on there. And if for whatever reason we can't get to a question, you can ask it in the group chat, and we'll ask the authors to respond to you guys there. So with that, I'll hand it back over to David.
Yes, excellent. Thanks, Grant. We picked two papers this week. The first of which is in ecotoxicology about seabirds, and the title of the paper is "In vivo accumulation of plastic-derived chemicals into seabird tissues." It's a publication led by Kozuke Tanaka, and it's a collaboration between Hokkaido University, Tokyo University of Agriculture and Technology, and Hawaii Pacific University. Today online, we are fortunate to have the correspondence author of the paper, Professor Takada from Tokyo University of Agriculture and Technology, And I think Professor Watanuki from Hokkaido University joined as well. So as you know, plastic pollution is a huge issue in the oceans, and it affects also seabirds, with nearly 80% of all seabird species ingesting plastics. And soon there are predictions that, you know, all seabird species will be exposed to plastic ingestion and pollution. But there has been a lot of debate about the actual impact of ingesting plastics beyond actual mechanical blockades. I heard a lot of people saying, well, you know, probably the stuff just moves through passively, and it does not contaminate the birds, or it would be the same rationale for fish. And there were correlative studies about contaminant levels in seabirds, but it was difficult to disentangle what comes from plastics and what through natural diet. So what the authors did with this paper is probably the first experimental investigation into plastic contamination. And they fed plastic pellets with flame retardants and UV stabilizers to strict shearwaters in Japan. And they studied the accumulation of these compounds in liver and body fat after 2 weeks and after a month. And when they compare the treatment with a control group, they found that accumulation of pollutants was 100 to over 100,000 times higher than the rate through natural diet. And through additional monitoring of preen gland oil in 6 seabird species from Hawaii, they detected these chemical additives only in those species with high plastic ingestion rate. So I think this is a really important study and something which had to be done. But of course, for the study, because the colleagues were working on tissues like liver and body fat, they had to euthanize the chicks. So that concerned 21 chicks. And I guess, you know, the question for the authors, which will be on everyone's mind, is there an alternative way to do this sort of experiments without killing the animals? For instance, the leading lab for this study developed a method using preen gland oil. And so my first question would be, would this be the way to go in the future?
Okay, and I'm just going to unmute the authors. Come on, buttons, there we go. And Yutaka, please go ahead, you're unmuted.
Yeah.
Yeah, hi everyone. I'm Yutaka Watanuki, coming from Hokkaido University. First, I feel very sad to do such experiment. We have to sacrifice first to know the clear result, how the plastic contaminant of the plastic come to the bird tissue. So I love seabirds, so I feel very sad to do such experiment. And but I now think that it is possible to do such further research. As David say, we can use preen gland oil or blood to know how the contaminant, the chemicals from plastic come to bird tissue. We will expand the experiment by using such a non-destructive method. And
Okay.
our experiment helps some types of new experiment.
Yes, and I also think that this was an important study at this time. Was there any particular reason? Can you hear me?
Yes.
Yutaka? Yeah. Was there any reason for choosing that particular species, the strict shearwater?
Yes.
Yeah, you pointed out in the method section that it's a really large colony, but I checked, and actually the IUCN status of the species is near threatened because it's declining. Did you have alternative species you could have worked with?
Yeah, why we choose strict shearwater is that the population size is great, very big, and very easy to access. And another reason is their feeding habit. For us, at this stage, it is very difficult to cage or keep the chicks in the laboratory at this stage. So we have to do such experiment in the field. And what we think is that if the birds feed on the plastics by themselves, it is a problem. We have to estimate how much a parent feed the plastics and give that to the chicks. With our information, there is no good published papers, but we know that the strict shearwater rarely feed on the plastics. So we choose that species. And this is, of course, the procellary forms that feed on many species feed on the plastics. So it can be the species can be a typical physiological response to plastics, but they don't feed. They rarely feed on the plastics. That's the reason why we choose that species.
Okay, and my last question would be more for Professor Takada, and it would be about the actual effects of the levels which have been measured in body fat and in the liver. What sort of physiological or maybe behavioral consequences do you expect at such levels?
Yeah, so far at this concentration, we didn't see any ecotoxicological effects on seabirds, but the chemicals have the nature to disrupt endocrine systems. So if the concentration would be higher, such effect may be seen. But at this moment, even at the concentration of this type of experiment, we didn't see any toxicological effect. But my colleague also measured the gene expression. He found some gene expression changed. That gene expression includes that gene regulates the synthesis of sterols. So
okay.
such kind of the ecotoxicological, the very slight change may occur.
Okay, thank you very much, and thanks so much for being with us online today. And well, congratulations on this really important study. And with this, I'm handing over to Grant if he has questions, or then we'll be taking questions from the audience.
Yeah, thank you.
Thank you very much.
Welcome.
So I just have a couple of quick questions. First one is related to body size. So if you have larger species, albatross or anything like that, are eating lots of plastics, is there a difference in the amount of bioaccumulation you're getting? Or would you expect there to be a difference in the bioaccumulation in the larger species versus, say, strict shearwater?
But I think the albatross ingests much more amount of plastics compared to the shearwater which we examined. So the ratio of plastic and natural food could be similar among the species.
Fair enough. And then, so if that's the case, then would you expect there to be more of a, if let's say, for example, you've got some of the smaller species, which may be eating proportionally more plastic than you would expect, you would sort of expect that would you expect there to be deleterious effects from the pollutants in the plastics, and what would those effects be? So what would be the impact on fitness, for example?
Yeah, you are right. Any animal, they eat much more plastics compared to their body size. The effect could be more serious. But to examine that point, we have to study much more wider species of seabirds and some other animals.
Mm-hmm.
Now we are studying such the effect by analyzing the 150 individuals of seabirds. We examined the Greenland oil for 150 individual seabirds collected from around the world.
Mm-hmm.
So we have to examine that effect.
Okay.
Yeah.
I do have one last kind of maybe nonsensical question, I guess, but it's just curiosity that's come about is so petrels and shearwaters are very messy birds. They like to puke a lot of oil and that. Have you ever come across any situations where the amount of plastic that they're ingesting actually changes how much they might be puking up in oil or how they produce that?
Oh, I'm Yutaka. Oh, I don't know. That stomach oil is a very important material that facilitate the chemicals come from plastic into the oil and into the stomach and into the body tissue of seabirds.
Yes, based on our previous experiment, the stomach oil can act as the organic solvent to facilitate the extraction of the chemicals from plastic. That, as Yutaka said, the stomach oil is the key of this phenomenon.
Oh, very cool. All right, I will open up the floor to questions now. So if anybody has any questions, just use your raise hand option on your screen, and then we'll go ahead and I'll unmute you. Anybody have any questions out there? Nobody's raising their hand out there. David, might you have something else you could prompt people with?
Yes, well, the other question would be what's the next step from there? So what would be the logical next steps in terms, of especially testing the actual effect of this type of doses or higher doses on the birds?
Yes, we have two studies. One is to study the actual toxicological effects by analyzing some gene expression. Another one is to know how widely this phenomena is occurring. So for this purpose, we examined the seabird, the Greenland oil, and we collected 150 Greenland oil samples across the world. And now we are analyzing the additives in the Greenland oil. So far, almost 40% of individuals accumulated the additives in the Greenland oil. That means this kind of transfer and accumulation could be occurring the global scale.
Yes, I think probably, well, I think there was a question on the forum, Grant.
I don't have any on my screen.
Okay. I think the question on the forum was that it was sometimes difficult to disentangle the effects coming from the environment from other effects. Was it? I vaguely saw a text flipping through.
Oh yes, yeah, I do see the question from Eliza in the chat room now. Yeah.
If you have access to the question, can you reread it?
Yeah, I do. Yeah. So Eliza has written, it's often difficult to isolate the effects of persistent organic pollutants in the wild because there is such a wide number of pollutants out there, and ecological variation means that pinpointing the cause of the effect is difficult.
Yes, that reason why we had to conduct the semi-field experiments. We added only 5 chemicals into seabirds to see their effect. If we would conduct such observation in the field, it's difficult to judge which chemicals could be contributing to any change in toxicological behavior of the seabirds.
And of course, seabirds, one of the characteristics of many seabird species is that they migrate over wide distances and also forage over wide areas at sea. And so orientation and also olfaction are really important, especially for procellariforms. Ultimately, do you expect any impact of contaminant levels on the capacity of seabirds to orient? Are there any studies in ecotoxicology maybe for other species which go into that direction?
Yutaka has
No, I don't know.
Okay.
David, you have any ideas?
Not really. It just came to my mind. I would need to check in the bibliography if there's anything in that direction. It would be tricky to test. You know, at the moment the test of contaminant levels, it's mainly on the impact on reproduction with knock-on effects on potentially population dynamics. But maybe I missed something, and there are studies ongoing about orientation. But it will be tricky to implement.
Yeah, very good comment. Thank you.
I've got a couple of questions from the audience here now. We've got one from Julian Beeman first. Julian, you are unmuted.
Hi, thank you. Thanks for having me here too. This is my first meeting, so thanks very much. I was just wondering, I don't really know much about ecotoxicology, but I'm an animal physiologist, and I'm just wondering, what are the main physiological targets for some of these disruptions? You know, is it to do with heart function, brain function, the nervous system, metabolism, thyroid function, or all of the above, I suppose?
Our concern by our study was conducted to demonstrate the transfer and accumulation of chemicals. So the actual toxicological effect is beyond our experiment. So we have to conduct some other sophisticated experiment in future.
What are some of your sort of, do you have any hunch as to what that might be? Do you have any expectations around the physiological effects, what they might be?
Yeah, I think the physiological effect could be seen much more later. Before that, some change in endocrine system could occur first.
Interesting. So, you expect that endocrine disruption is really the first major.
Yeah. Because those chemicals, BD209 and UB327, is a kind of endocrine disrupting chemicals. If their concentration could be much more higher, such effect could be occurring. But during our experiment, we didn't see any the
Sure.
obvious.
Sure. Thank you.
Thanks, Julian. All right, we've got another question here from Chris and Karen. Go ahead. Go ahead, guys. You guys are unmuted. Nice to see you, Karen. How you been?
Oh yeah, nice to see you guys. This is just such a great forum, and I'm really excited to take part. So my question, I understand that we're still trying to get a handle on toxicological effects, but one of the things that I'm really interested in is the fact that seabirds are a really great indicator of what's going on in the ocean. And it seems they're also an indicator of the fact that if there is increasing plastics in the ocean, they're also potentially picking up plastics at a relative rate to what the increase of plastics or potentially decrease of plastics might be in the ocean. So I'm just wondering whether the amount of chemical that you can detect from plastics in the preen gland oil might be something that we can use to indicate changes in the amount of plastic that is coming into the ocean. There are lots of different indicators that are being discussed, but I'm just wondering if this might be something that could be used in the future, if we could use that to measure change over time. Does that make sense?
Yeah, it's a good point and a good approach. But the problem with additives or chemicals in plastic is their occurrence is very much sporadic, not uniformly contained in plastics. So it's so difficult to judge the amount of plastics by using some additives in the Greenland oil. So very much product. If we measure some chemicals in 10 plastic fragments, we encounter only one fragment with very much high concentration of the additives. The other 9 fragments didn't contain the plastic additives. Therefore, by using the additives, it's difficult to judge such effect. Also, that means that we have to measure larger numbers of the individuals to see the effect of the plastic additives. And thank you for your comment and idea.
Thanks a lot, Karen. All right. Is there any more questions from the audience? If not, then we'll move on to the next paper. I don't see any. All right. Thank you to the authors. That's a lovely paper you guys have put together. And thank you very much for joining us. It's very appreciated.
Yeah.
Thanks a lot.
Okay. All right. The next paper we're going to be looking at is kind of a cool one, actually. I mean, all the papers we look at are cool. What am I talking about? This one here is actually one that I followed when it came out relatively recently. I'm a bit excited about it. I'm happy to be the one to introduce it this morning. So we all know that a common pitfall of most attempts to quantify species in space and time is that we sort of lack an understanding of how distributions differ between life history stages. And though, you know, we can track adults and we can track juveniles, there's still a lot of questions that remained around that. You know, basically juvenile and adult seabirds may not be using the seascape in the same way. And of course, as we've been tracking more and more birds, this has become much more evident. So this paper that we're chatting about, the second paper we're chatting about today, comes from a team led by BirdLife International. And it's got quite an impressive authors list, a lot of heavy hitters on here. And David, I know you're actually a co-author on this, and I'm not just trying to butter you up. This paper basically describes a method that integrates tracking data from 22 species of procelariform seabirds with phenological and demographic data. And they do that to infer distributions for various life history stages. So I was excited about this paper because it really starts to fill in some of these key gaps in our understanding of spatiotemporal distributions. The authors point out sort of misdirection of management and conservation efforts can be very likely without taking into account life history stages. So an example of this might be in the Antarctic community. For example, a lot of the latest work looking at the overlap between penguins and the krill fishery doesn't really take into account the distributions of various life history stages, and particularly fledgling penguins. So on the Antarctic Peninsula, when the krill fishing industry start moving in sort of in late spring or late autumn into their early in their winter, that's sort of when the fledgling penguins are specifically chin straps, for example, are in the water for the first time, and we have very little information about their distribution and the overlap of those two of the fishing industry and the young penguins. So this paper sort of outlines a broad methodology for which we start to estimate these distributions. It starts by first estimating the proportion of the population in various life history stages using modeling techniques. Then they go about calculating utilization densities from tracking data, and they classify these by parameters, or group them by parameters like breeding site, the age, type of device, and this is called a data group. So these data group utilization densities are combined and weighted based on things like the phenological data and life history stages. And then those distribution maps can be aggregated in time and space to various management units, whatever might be necessary for the specific question. So it really outlines this really nice methodology, and they've produced some R scripts. There's an R Shiny app web application that's associated with it as well, which I checked out last night, which was quite nicely designed. So the population models that they developed for this suggest that for many populations, breeding adults generally represented less than 50% of the total number of individuals, which is quite interesting, because assuming that this is correct, then obviously studies that only track breeding adults are unable to generalize across the entire population. So that raises the question, whether or not we should be focusing more on tracking juveniles as well. The other interesting result that came out of this was the overlap with the pelagic longline fishery. Basically what they showed was that if you're using only breeding adults, there was an underestimation in the amount of overlap. And a highlighted example of this was with the black-browed albatross. And they suggested that by including juvenile and immature distributions, the exposure of the birds to longline hooks was increased by an average of 1.4 million hooks. So that's quite a substantial change. So I do have a few of the authors on here, and I have a few questions for them, and I'm just going to unmute them. Let's see, here's Anna, and Anna is unmuted. I wonder, do we have anybody else on here who was on that paper other than David? Yeah, David is here.
And Maria and Lizzie.
Oh, are they here as well?
Yeah.
Yeah.
Okay.
Lizzie was responsible for the Shiny app.
And Lizzie, where is she? Here we go. Lizzie, unmuted. All right, cool. So I've got a whole bunch of you guys online at the moment. Great, and thanks very much for this paper. Like I said, I was really excited about it. These issues are near and dear to my heart. And so one of the things that came out of the paper was obviously that tracking juveniles and immature birds was important, because they make up a relatively large proportion of the population. In seabirds, it's kind of, I guess it depends on the species, but in general, it's kind of understood that the adult birds are sort of the important recruiters into the population. So when it comes to overall protection, does it really matter that we're to track juveniles and immature birds? What kind of proportion of, what are they bringing to the table, sort of, and should we really be focusing on them, or should we be focusing more on the adults?
I think there is still a lot of unknowns here. So for a lot of species, we don't really know exactly where the juveniles and immatures are going. Some of them, they do segregate quite a lot from the adults. So the ones that we have some data, so it is important. And I think as well, it is true that, let's say that adults will potentially count more, but what happens is that some populations, we know that like recruitment is really low. Like a lot of populations in South Georgia, you know that a lot of, adults are doing fine, but you know that the juveniles are not recruiting back on the population. So I think we need to pay a bit more attention for these classes. Because yeah, it might be that if they are segregating a lot from the adults and they are going to areas that potentially they can interact more with fisheries, maybe the adults are doing fine, but there will not be juveniles in the near future recruiting back in the population. So eventually we'll have a crash, as we can see a lot with a lot of populations. Like South Georgia, this is a major problem.
Do you see this as being this is a species by species issue, or do you think that this could be something that's generic across all species?
I think for a species that you see more segregation between juveniles and immatures and adults, then yes, it's a kind of a species by species. But we don't really know enough, right? There is very, very few data on juveniles and immatures. And I think the idea of this paper is actually to present the methodology and encourage people to collect more of this data and hopefully like run this and that's why all these scripts are available, because the idea is like for the scientists to go to the field, collect their data, and make the sample sizes of their own species populations like much better, run these scripts again to see what is really happening with their populations, because we know that we don't have enough data.
Very cool. So following up on that, with regards to sort of figure 1 of your paper, you show these really lovely breakdowns of the age structure for various species. And I'm wondering, was there any way of ground truthing those estimates, or were any of them ground truthed? And how realistic do you think those population breakdowns were?
Well, we asked a lot of support from the scientists that were involved in the project. So we got a lot of feedback from them in terms of, and I think people that are working for like 20 years with that species, I think they have a quite good knowledge of what is going on. So I think, let's say that my ground truth was more like the opinion of the researchers.
Expert opinion.
Yeah, can I add something also on that? Maybe using the example of the black-browed albatross, I think another something that came out from our study was precisely bringing the juvenile and immature data. We saw that there's a lot more distribution, let's say, in Brazil, in areas that was for some time something that our colleagues from Brazil, from Projeto Albatroz, they were asking us to help them, because all the maps were showing that black-browed albatrosses were not going so further north, but they had a lot of bycatch, especially of young stages. And it was difficult to justify with the government that albatrosses were there, because most of the maps were based on adults. And basically, and that's why also the reason why we chose the black-browed albatross, because I think it's a good example where bringing juveniles data and immature data can in fact help explaining something that we knew from other sources of data like bycatch, but was not possible to show with maps that we had before. So is a kind of not exactly ground truth in terms of distribution, but helps explaining some of the bycatch of albatrosses in South America. And also, sorry but also linking to the previous question, something that as Anna was explaining, the idea was to build this framework. But also the utility of this, if, for example, if we get more data on the bycatch rates of different stages of seabirds, we can in fact improve this kind of analysis by taking into account possible differences in the bycatch rate of juveniles, immature, and adults, because there's some evidence also that some stages sometimes are more bycaught in some species because maybe the juveniles or immatures are more naive or whatever, but that can be also different between life history stages. And combining with this kind of maps, we can see a much better picture of what is going on in terms of bycatch and other threats, of course.
Mm.
Can I just quickly jump in on the demography point as well? I think that's a really good question. And what we were really trying to do here is find a model that we could use for all of the species. So obviously there was a trade-off between making it like the best model we could and making it applicable to all the species. So we had to kind of find something where we could actually find the demographic parameters for all of those species. But really, with the framework that we've made, if you're doing it for a smaller number of species or specific species or populations, you can switch out our demography model for a more specific demography model to your species. Once you've got those breakdowns between the different life history stages, then move on to the framework in the way that we've presented it. So really kind of, we're trying to make it quite modular so that if you do have more demographic information and better demography models specific to your species, you can use those instead of ours.
Very cool. Very cool. So just out of curiosity, it was quite interesting looking at that figure. There was the models seemed to suggest that there was quite a lot of variation in the breakdowns of the age structure. And even for species that seem to have somewhat similar life history strategies, is there anything that accounts for that in the models? Like what specifically would lead to that kind of variation? Or is this something that you think is actually reflected in the populations?
So I think a lot of the variation came from the species being annual or biannual, and this is accounted in the models. Yeah, so it is accounted in the models, and I think potentially maybe these models can be improved by like looking at if you know that parameters will have a kind of very big confidence interval, you could potentially do a sensitive analysis in order to try to make sure you're actually, if juvenile survival varies like a lot, you could potentially do sensitive analysis with confidence intervals. We've done something more general here, but I think the majority of the variation in terms of being annual or biannual, they are already captured in our models.
Yeah, very good. I gotta say that's quite fascinating stuff. I have one last question, then I'll hand it over to David to ask his co-authors some questions. But my last question has to do around the resolution of the outputs being at 5 by 5 degrees. So obviously that's quite coarse, but in the paper you talk about how this can be scaled to various resolutions. And I'm wondering if you have maybe a couple of examples of what kind of spatial resolutions might be appropriate for specific circumstances.
I think it depends quite a lot of your question and the data that you have, right? So here we had the data on fishing, which is reported by a lot of RFMOs, like regional fisheries management organizations. They are reported at 5 by 5 scale. So we thought that for our use, for us to go to the conferences and to these meetings, we have to have the data on this scale. I'm not saying that this scale is the ideal. I think if you can go like finer than that, probably it might be even better. But I think a lot of times we are quite limited on the data. So I know that, for example, there was a lot of plastics that are like in 1 by 1 scale. So I think potentially you could do that based on 1 by 1. I think the resolution of the depending of the tracking devices that you have as well. So I think it depends a lot what threat you are looking at.
Very good. Thanks a lot. David, do you have any follow-up questions for your co-authors? I know you're probably entrenched in this paper already, so you may not have very much.
Yeah, I'll probably stand back a bit and we'll be taking questions from the audience, but a tiny one maybe. In the paper it says that it's also applicable to areas with not so much information, but you do need some baseline as information. After running the study, do you see really major dark areas or areas, for instance, of the Southern Ocean or from some group of species where there's clearly lack of information, and a big objective for the next few years would be to perform more tracking, for instance?
I think a lot of juvenile and immature data. We are missing a lot on this life history stage for sure. So if we could collect more data on these stages would be great.
Yeah, and talking about species, that probably concerns also smaller petrels.
Yeah, no, for sure. Maybe Lizzie, that looks a lot in the Seabird tracking database. Would you have another suggestion?
I'm not sure of any particular species, but yeah, I think just juvenile and immatures particularly. And also some of the data we found quite difficult to get hold of was also the demographic parameters data. That's quite difficult to know, to find data that's representative of the species over quite a long period of time, because often there's just a few studies or from particular years and never quite sure how big the error bars are on that. So yeah, some of the demographic parameters as well, particularly for less studied species, but yeah, really tracking data for juveniles and immatures.
Okay.
Yeah, and if I can add, going a bit outside of this paper that was really focused on albatrosses and petrels and also linked with the problem of bycatch. But well, in my opinion, a big gap in terms of knowledge of small petrels and storm petrels, and because we have all this general idea that the problems with these species are only in the colonies, and maybe they are, but the reality is that we have very little knowledge yet about the distribution of these species, and because they are so difficult also to study in the colonies and to know the trends. I mean, it's maybe possible that they are also declining a lot, as many albatrosses, because they are so, difficult to study, mostly nocturnal. We don't know. So yeah, it's storm petrels. I would say it's a big gap in terms of knowledge, in terms of at-sea distribution.
Yeah, that's very good, and congratulations to you guys on a really lovely paper. This is obviously a big global effort between a lot of co-authors, and it's quite an achievement to get everybody to agree on a paper.
Yeah.
All right, I will open this up for questions from the audience. Does anybody have any questions out there? Please raise your hands on your screen. You can throw stuff at your screen if you've got your camera on, or wave your hands frantically. I might be able to see you. Surely someone has some sort of interesting question out there for these folks. It'd be a shame to let everybody go so soon. Oh, Karen looks like she's got a question. I'm seeing her waving her hand at me, so I'm going to unmute Karen. All right, Karen, you're on.
Oh, hi, and a special hello to Maria. This looks like absolutely fantastic work. So I'm of course thinking to the practical application of this information, and I'm thinking that there's a couple of things I wanted to ask. So, for example, in your figure 5, you've got some very clear high risk areas for overlap of fisheries with albatross distribution. And I'm just thinking, have you had a look, or has anyone had a look at the actual bycatch data to see if you can actually verify, what you're seeing here with the bycatch data? And then I'm thinking you can then take that to the next step where you can start potentially advocating the use of this information to get potentially spatio-temporal closures, or at least restricted areas, because it's very specific in terms of the actual longline fishery. Is that something that you're potentially thinking about?
So in terms of looking into the bycatch data, I think these are kind of complement a bit of what Maria was mentioning. So we looked, there was a lot of papers and reports from people from Projeto Albatross mentioning a very high bycatch hotspot in the south of Brazil. And I think for the first time we could actually see that the birds are going there based on tracking data, because before, using just maps from the adults, we couldn't see that, so it was really hard to advocate in terms of that. So having these maps, I think potentially we open kind of a new avenue in terms to try to do more advocacy work. Yeah, so people, for example, from Projeto Batros, they were extremely happy with these results because they think that, okay, now I can go to my government and try to do something, because every time we arrive there with like the general maps, well, the government, they just say, well, but the albatrosses are not here, because yes, we tend to use a lot of tracking data just from the adults. And I think this is the first time that they actually were like, okay, now we are happy with this, we can go ahead. So yeah.
Right. And can I also add that, I mean, and thanks to Karen, that was, I should say, and to explain a bit maybe the very beginning of all this project and idea of doing this framework and publishing was a lot of Karen's work, and also other colleagues from BirdLife that work in fact more closely with the regional fisheries management organizations and the governments, the Albatross Task Force, that maybe almost every year we're asking our marine science team to do these kind of maps, and was always a kind of, because it's like a rush and compiling all the data. So we thought, okay, let's create a framework to be more standardized and consistent and help creating these maps precisely to have this final aim of doing all the advocacy work to reduce the problem of bycatch. That was the big motivation. So thanks, Karen. Because yeah, that was the very beginning of this idea. This paper, but it was a very practical question to help solving the problem of the albatross.
Okay, cool. Well, best of luck. Good luck.
Thank you.
Great. Thanks a lot, Karen. Do we have any other questions from the audience this morning, or this evening, this afternoon, whatever time it is in your part of the world? I don't see anybody else raising their hands. David, do you have any other statements you'd like to make? And then I think we can let people go on their merry way and back to their lives.
Yes, I had some feedback of people asking the reason of using UDs to define the at-sea areas. That's an ongoing debate. So I think people wanted to know whether there were practical reasons for choosing UDs, whether there would be alternatives which would be workable.
Yes, there is a lot of debate about that. Let's say that we try to, I think the way that we calculate our UDs, well, I'm not entirely sure I can say it's better or not, but so we try to take a lot of things into account. For example, so we calculate a UD for each single individual, and then we average them towards the end in order to kind of account for different lengths of the trips. So I think we were quite careful in the way that we use the UDs. And in the way that we use the UDs as well, it's something relatively very similar to what would be like time span grids, because we didn't, yeah, I don't know. And I'm not entirely sure how to answer, like, well.
Can I give also my opinion?
Yeah, I have one also, but go ahead, Maria.
No, it's always a bit of this debate, what the best method to do, this kind of analysis, and we know that there are already a lot of different methods out there, maybe most of them or many of them much more refined and detailed. I think two important aspects to take into account. One, we want something that was relatively simple, also because of the reason that I was saying before. In most of these policy discussions, people want to, very complex models. Sometimes it's much more difficult to sell them for people that make decisions, because it seems that it's very obscure and a kind of a black box that they don't understand so well. So it's important to have a simple method that is more, it's not so complex in terms of analysis, and kernel analysis is relatively simple in that way. Second is the spatial scale that we are talking about. Because we are not looking to the detail of the individual behavior, right? We are looking to something a much broader scale. And even if we go finer than 5 by 5, it's still, to do conservation, we don't do conservation at a very individual level. It's something that it's a much bigger scale. And to be completely honest, and we did some tests, the difference between doing very complex things that look much more into the detail of the behavior or to do something more simple, but like a kernel, is very little at that spatial scale. So we prefer to keep it simple to be also easier to, let's say, sell to the more advocacy levels.
Very cool.
What is your opinion, David?
David, yes, now I'm kidding.
I think you summarize it very well. You know, people get very excited about analysis and the different methods. And my personal experience is that when you look at it like a decade later and think, you know, how excited people got and had really strong opinions, it didn't change radically, you know, the biological facts. That's probably my opinion. So it's always really important to look at refinements, but as you mentioned very clearly, when you're working with conservation, you know, you have to be very strategic on how you spend your efforts. So there's a notion of efficiency, I guess.
Mm-hmm. Yeah, and that's right, and that's sort of why I like the way that the paper is really well laid out. This nice methodology for going from A to B sort of speeds up the process. I like the fact that you guys have integrated this Our Shiny app, so you can get the population structure metadata out in a format that the code can accept. So that's quite slick. I'm excited to see where that goes in the future. So thanks everyone. Thank you to all the authors for joining us today. It was really, really great to have you here. It was great to discuss these papers. I'm looking forward to hopefully seeing all of your faces here again in the coming weeks as we continue this series. And yeah, congratulations everyone. And thank you for everyone who's tuned in today. I know we're still trying to get our feet on the ground in regards to how the scheduling is going to work with this. I think we've sort of struck a bit of a sweet note at the moment with regards to the timing. So if anyone here has any other questions, please raise your hand. If not, then I will sign off and let you all get back to your real life. I see that my daughter has shown up in one of the screens.
It's not a question, but I just would like to say that, well, that's great organizing the section. So thanks for the initiative. It's really nice.
Thank you.
Thanks a lot for being with us.
All right, everyone. Take care now. Stay safe, wash your hands, stay indoors. Bye now.